Hello, our talk is entitled An Introduction to Modern Computational Biology Through Microbiome Research for High School Students. My name is Josh Kangas, and I'm an assistant teaching professor in the Computational Biology Department at Carnegie Mellon University.
And I'm Philip Koppa. I'm an associate teaching professor in the Computational Biology Department in CMU School of Computer Science as well. And so we wanted to talk a little bit about our program today. Just for some background, we teach a 3-week-long full-time program for high school students. They come in from around the country, and we've even had a couple of international students. It's a fee-based program, but it's very competitive in terms of our applications. As I said, students come from all around, and we fit in with an infrastructure, or an ecosystem, I should say, that CMU provides in terms of almost a dozen programs that are aimed at rising seniors in high school, whether it's drama or architecture or video game design. And so computational biology is one of these programs, and we're proud to be a part of the CMU Pre-College experience. Our students, we didn't know how we would fit into this year one. So we started this program in July 2019, and we had a target that we wanted a couple dozen students. We didn't know if we would reach that. And we were very happy to find that we had absolutely superlative students. We actually essentially closed our application early because we reached a couple dozen students who were very excited about mathematics and bio. We did not require that they had any computational biology background before they came to us, nor did we require that they had done any programming before they came to us, as we had a short boot camp on programming as well as part of our program. So we wanted to talk a bit about the program, kind of how it is set up, what we wanted to do. And I think a large part of why we were very quickly popular is that we structured our program around doing not just a toy research problem with students, but a real research project that had not been performed, which was studying the microbiome of Pittsburgh's 3 rivers. And we were interested in working with our students to kind of figuring out what drives the variation. Is it weather or seasonality? What about different locations within the rivers? As well as the fact that we have an opportunity to study the confluence of 2 rivers in Pittsburgh to see what happens after that point in terms of the geography of the rivers. So we have big picture goals for what we want to do with our students. First and foremost, and which is why we've put it at the very top there, is we want our students to have an academic experience that is fun and that uses active learning, which I'll talk briefly about shortly. We have a very strong, we want to have a very strong demand that students get to sample a real biological data set. They collect all their samples, and then they do an end-to-end analysis where they apply the techniques in the laboratory, and then they design programs that will solve computational problems and then apply that to the real data that they gather. So as part of that, we want them to appreciate that in real biological research, computation is ubiquitous. A sub-aim of that is that we want our students to write hundreds of lines of code on their own that's going to be wound up applied to analyzing biological data. And then the final point is that we want this to be a challenge. We don't give grades. We don't have we don't have individual assessments. The goals are big picture. The goals are to have an experience that students really cannot get anywhere else in the world. That's outside of the standard high school biology or computer science curriculum, and to appreciate that real science is about challenging oneself, and we want our students to feel that challenge, especially because so many of them are such superlative students that they don't probably get this challenge often during high school. So how we have structured this is through active learning, meaning having students do as much as possible. That's certainly the case in the lab, where they're collecting their own samples and then they're applying lab techniques in order to capture biological data. But it's also true when downstream of that, we have them kind of work in groups to do short code challenges, come back together, give them the next code challenge, and have them do as much of the programming on their own with TA and instructor guidance as possible. Getting student buy-in to this is really important, understanding that we've spent a lot of time structuring it in this way to make sure that every moment includes active learning. And so we point our students to real research results showing why we've added active learning to our experience. So we point to, for example, one PNAS paper from 2014, Freeman et al., showing that active learning boosts student performance by about half a standard deviation in STEM courses that have grading, as well as showing students that they're going to be more challenged, and they may feel as a result of that they aren't learning as much as they might in a lecture-based classroom, where it's easy to follow along and smile and nod, but you're not constantly tested on your knowledge necessarily. And so pointing out that this is real research from just 2019, where students have the sentiment that they're not learning as much, while meanwhile they are actually performing better than they would in a traditional lecture. So that's the setup for kind of a big picture of what we want to do. And Josh is going to talk about some of the details of how we set up the research program and what we found with our students. So take it away, Josh.
So we started this research program by applying for a small grant, and we received that grant, and we used that grant to fund this program and this research program across different programs. So we have an undergraduate program, and we actually incorporated this microbiome research into a fall 2018 class. This is a picture of a couple of our students out in the Ohio River taking water samples and then processing them. And then here, this is our winter 2019 sampling. And in this case, students weren't too interested in sampling in January, and so it was just Philip and I doing the sampling. We were clearly enjoying it. And then in this one, this was in May, but it was an absolutely frigid day, and the students were more interested in studying for finals than joining us on this excursion. So we got samples for the spring, and then in the summer we had the pre-college program. So this is a picture of all of our students before we boarded this vessel behind us. So this is Rivers of Steel is a nonprofit organization that runs a science vessel in the Pittsburgh area, and so they took us all around the rivers where we could collect samples in the locations we were interested in, and it was a very nice trip. Okay, as far as the specific locations, we chose these locations because they would allow us to test for the effects of the seasonality. Because we had different locations, we could also test for the effects of different locations, and also the confluence of the rivers. And then finally, as we were going along, we also collected data on water conditions like temperature and pH. All right, as far as the actual processing of the samples, so we filtered the biological material from the water, and then from those filters, we were able to extract the DNA, and then we sequenced that DNA with various methods, and then we analyzed the sequenced DNA using algorithms the student built, and they were building these in the computational lab, obviously. Okay. All right, so this is an overview of the experimental process. It's important to note that right at the beginning of the program, the students came in, and in the computer lab, they received a programming boot camp where they learned to program and Go. And then in the wet lab, we did a pipetting boot camp where they learned, the basic skills that were needed for both kinds of labs. These are the specific activities, and I've broken them out here into different categories. Next slide. So first we isolated bacteria and then did different tasks. So I'll tell you what we learned from these, and next slide. So for the isolated bacteria, in the computational lab, we taught students about sequence alignment, and they actually implemented alignment algorithms, and then we showed them BLAST. And so we did Sanger sequencing on bacteria that they isolated from the water samples, and we found, we identified 20 species that we isolated. Next slide. Then we also did whole genome reconstruction, and computationally, they implemented an algorithm for that, and they also did ORF finding and gene annotation. And what was really interesting here is that one of the species of bacteria that we isolated was called Flectobacillus roseus. It had never actually been fully sequenced before. It was only identified based on its 16S gene. So we actually generated the full sequence of that particular bacteria, and we're in the process of publishing that genome. Okay, then we also did some tests for antibiotic resistance, and this gave us an opportunity to do basic image analysis with the students. And just one basic result that we have that's obvious here is that this particular bacteria, Bacillus aryabhati, is resistant to sulfamethoxazole, as you can see in this image. Then we did analysis on microbiomes. So we're looking at whole populations of bacteria from the river water samples. Computationally, we did alpha and beta diversity analysis. We also did principal coordinate analysis. And from a conclusions perspective, this plot is showing the primary conclusion, which is that the diversity of the microbiome is primarily driven by seasonality as opposed to location, which is not a huge surprise, but we were able to demonstrate the analysis to come to that conclusion with the students.
So a very important part of all this was that we wanted to make sure that students then had a chance to reflect on what they had done. So as part of the 3-week program at the end, we had a final student presentation to the students, parents, and families and guardians that lasted 3 full hours. So we wanted to make sure that every student had a chance to speak for a few minutes, and they outlined end to end exactly what we had done for a lay audience over the entire 3 weeks and compressed that down into 3 hours. So it's just a testament to how amazing our students were, and they were just as enthusiastic at the end of the presentations as when we had started. And the other thing that we did was we asked them to tell us what they had learned. We thought that was the best thing, as well as for venues like this to explain, kind of, we had these big picture goals, but what did students feel like they learned? And we were expecting them to potentially give us some low-level answers, like how to use object-oriented programming if you're building a genome assembler, or something, how to pipette effectively and how to carry out kind of a certain aspect of some of the lab experimentation that we did. But we were amazed that in 5 minutes, some of the things that our students said, that we would have been impressed with kind of a PhD holder being able to reflect on the nature of computational biology in such a way. So I'll just quickly read what they said, because I think it's the biggest kind of keystone of our program. So biological data are messier than you think they will be, and that's not a problem. Heuristics are very valuable when dealing with real data because our problems aren't solvable by exact means. No model is perfect. Some problems may have a perfect theoretical solution that doesn't work at the scale of real data. Parameters are vital when building computational approaches, as we all know in bioinformatics, and they need to be tailored to the data set that we are using. Fundamental topics of computer science like dynamic programming, recursion, object-oriented programming, et cetera, are everywhere in practice. That was a great one, made me very happy. We often have a trade-off between the accuracy of an algorithm and its precision, or between the accuracy of an algorithm and its speed. I mean, that's a very high level when computationally that we were very thrilled that they saw. We need precisely formulated computational problems in order to answer biological questions. Biology and CS aren't distinct fields, and there are major opportunities for people who can understand both. That's a great one for CompBio as well. When solving any problem, we should focus on the big picture first, and we can later zoom in on details. And finally, often biological research is indirect. So we have to answer questions about things that we cannot directly observe, or maybe never will be able to directly observe. So normally that might be how we would end our talk, because we were so happy with them being able to reflect on that. But Josh is going to talk a little bit about kind of how we've torn down the program, especially in response to a pandemic, but also with respect to a few other things as we look toward the future.
Okay, so for the pandemic edition of the pre-college program, obviously we had to make it an entirely remote program. And so one of the things that allowed us to do was to double the size of our program. So we're almost double it. We're up to 51 students. We also saw that the students did very well in the last summer's version, but we wanted to give them an opportunity to do more. And so we gave them the programming boot camp beforehand. So they did that as prep work before coming to our program. And so far that seems to have been a great success. Unfortunately, we can't do lab work because it's remote. And so we're using the data from last summer with the summer students. We've also improved the program by making a lot of the programming tasks more group-based, and so the students are actually very actively involved in doing the programming in small groups with each other. We also have more TAs, so we have 5 TAs this year, and that really helps to improve the interactions between the students, and then the students get a lot of attention from their instructors. And then yeah, the coronavirus definitely provides us an opportunity to teach students about an active research area that has enormous relevance to the world right now. So it's been a great opportunity for that. Another part of this that's important to us is that we're consistently trying to improve gender parity and diversity. So this year we have actually added a number of scholarships as a result of a generous donation to the CompBio department. And we've improved our diversity, but there's still more to do. And then in the future, we have a number of improvements we intend to add to the program. So one of them is that CMU has definitely a strength in automated science. So I actually have a robot as my background that we may be able to use as part of the program in the future. We're also definitely working with partners within the university and in the local community to help us improve the diversity and gender parity of our program. And then we're always looking for real research topics that we can tailor to a level that high school students can handle. And that means both within the lab and in terms of the computational analysis that those projects require. So to close, we have to acknowledge all the people that have helped us with the program directly. So we have all of our TAs from last year and during the current program, and then we have Sam Madrinich, who's our program coordinator. We definitely couldn't have done this without her either. And then ultimately, we do this work because we enjoy the students, and we also really get a lot of benefit out of teaching them. And so if not for the students, we wouldn't be doing it. So we give our thanks to them as well. All right, and we want to thank you for listening to us.